Hi, everyone. Welcome to our expert panel on PAH. It's my pleasure to introduce you to Dr. Bhuvin, Director of Pulmonary Hypertension Program and Co-Director of ILT Program at St. Joseph's Medical Center in Arizona. And today we will learn about the treatment paradigm in PAH and how emerging therapies are set to transform it. I have here with me Martin Voss, CEO of Tiakis Biotech, and Rahul Agrawal, CMO Cereno Scientific, two biotech companies advancing some exciting programs in PAH. Martin and Rahul, please tell us a little bit about your background and also your companies. So Martin, you want to go first?
Yeah, sure. First of all, thank you, Debanjana, for inviting me to this call. I really look forward to our discussion today. By means of introduction, my name is Martin Voss. I'm the CEO of Tyakis Biotech, and I have background in corporate finance and M&A advisory. Our company, Tyakis, is based in the north of Germany, and together with my 10 colleagues, we are working on the clinical development of our neutrophil elastase inhibitor, Treprostinil, a recombinant copy of the human endogenous protein called elephant. And we developed this protein as a differentiated treatment option for pulmonary arterial hypertension. In PAH, that's a closing word, we are closely collaborating with Stanford University and are currently preparing for a worldwide phase 2 trial in PAH. And towards that end, are also raising a Series B financing round of $60 million.
Thank you. Rahul?
Thank you very much. And it's a pleasure to be in this panel. Thank you very much for the invite. My name is Rahul Agrawal. By training, I'm a cardiologist, and currently I'm the Chief Medical Officer and Head of R&D at Cereno Scientific, which is a Swedish stock-noted company. We have 2 clinical assets in the development and 1 preclinical asset. Our goal and vision is actually to pioneer treatments to enhance and really extend the lives of patients and people, I should say, with rare cardiovascular and pulmonary diseases. And there we are working on epigenetic modulation. We believe we have some innovative approaches to really tackle the root cause of the disease. And there we are on a very good path in this direction. We're working and collaborating with several US sites, with Abbott, and University of Michigan as well. And it's a pleasure to be here. Thank you very much.
Thank you. So before we dig deeper into your assets, maybe I would like to know from Dr. Buddhdev how more about his practice settings and how many patients he's following annually.
Yeah, so I started the pulmonary hypertension program along with our very well-established lung transplant center. We're following roughly about 250 to 300 PAH patients. They're group 1 is what we put our database in. And then we have roughly about over 100, 120 PH-ILD patients, and then the rest of the groups in the other mix.
Sure. And what's your current approach to treatment sequencing in PAH?
Yeah, we're still strictly following the WHO PAH symposium guidelines that were recently published, as well as ERS ESC guidelines. We start out with dual therapy. As we knew, dual was same as triple therapy. We start typically with Opsinvi at this point. We do like the combination setup where we're 1 pill once a day, 1 authorization. We start off with that. We follow them in about 6 to 8 weeks. We'll do their reveal light scoring at that point, and typically add a prostacyclin if needed. Now, these are patients who are class 1 to 2-ish or 3 when we are starting out their therapy. If somebody's class 4, we're going with parenteral prostacyclin along with Sotatercept and all classes of drugs at the same time. So again, it's very individualized, but that's kind of the general treatment paradigm. And once we follow up, adding prostacyclin, we recognize that these patients are the severe phenotype. Then we look at Sotatercept right at that point and start authorization process for that as well.
So besides patient severity, are there any other factors that shape your treatment decisions, like patient preference for modalities or?
Yeah, there's some preference in terms of parenteral prostacyclin for sure. I have had patients who absolutely do not want parenteral prostacyclin, and now having sotatercept in the mix has helped us reduce the use of that to some degree. Previously, we would have somebody on triple drug therapy with oral or inhaled prostacyclin, and if we are switching them to parenteral, that was our only option as they were getting sicker. But now we add on sotatercept and not have to have them go on parenteral prostacyclin. Other than that, there's not a whole lot in terms of preference. There are people who obviously do not like inhale 4 times a day, or we would have somebody who has a lot of side effects with oral selexipag is what we use a lot of times. And in that case, we would choose other mode of use for prostacyclin.
So you brought up Sotatercept, of course. So it's probably one of the major advancements we have seen in the field.
Yeah.
What's your take on the long-term safety and efficacy of the drug?
Yeah. So we have had several patients to about 20 to 25 that we started right off the bat in the Sotatercept group, which were carefully selected. I think we have been very careful in who we put on Sotatercept. So these are class 3-4 patients. It's usually an add-on therapy to the current 3-drug regimen. And they don't have portopulmonary hypertension, so they are not thrombocytopenic to start out with. They are not on a lot of antiplatelet or anticoagulation along with, so that the bleeding risk is lower. And with that, we did not have any major side effects so far. I had to stop sotatercept on one person only because of some GI bleed, but out of 25, they were tolerated well, and we had to reduce dose on one. But other than that, we did see telangiectasias, we did some hemoglobin increase, some platelet drop, but they were all very self-limited.
So besides these safety issues that you probably have encountered, are there any reimbursement challenges that you're facing with Sotatercept?
Not really. I mean, my nurse practitioners and our team, they handle all the insurance authorizations and pre-auths and all of that. And we haven't so far had an issue where it was completely denied. Every medication just requires 2 or 3 attempts to get it and reimbursements and things like that. But it hasn't been an entire denial by any means.
And so now that you have all these approved drugs, what would you say are still the biggest unmet needs in the space? And what do clinicians want to see from the next novel therapy?
Yeah, I mean, I think we're really focusing on the disease-modifying concept now. We obviously have improved the mortality in PAH from, about 3 years when we started out about 20, 25 years ago to now we're about 10 years or more. Unfortunately, we're still stuck to the initial regimens that we started out with ERA, PDE5, which are, they're vasodilators. They improve the heart function for a period of time. They improve the markers, and then obviously the disease progresses as we know of its trajectory, and it's only a matter of time that patient will eventually need a lung transplant or pass away. And along the way, we kind of, let's say, prostacyclin, and now we had sotatercept and all of those. But I don't think we are really targeting the core of the disease. So a couple of things. One, we are trying to learn if sotatercept could have use a little earlier if it is a truly disease-modifying agent, and we are looking forward to new disease-modifying agents who are really targeting the disease itself.
What would you say are the most important clinical endpoints for physicians and patients?
Yeah, I think for patients, they typically look at their functional status. So they are very focused on their 6-minute walk distance, and that tells us directly if they are, it would reflect if they tell me I'm doing better, it would reflect in their walk test. For clinicians, we obviously look at cardiac BNP. We look at the echo, every 3 months, evaluate and make sure the RV is actually reflecting the improvement as well. So overall, we look at every 6 to 8 weeks when we are following them, we look at the reveal light scoring, and that has to improve for us to say that we are making a difference. And lastly, we always really focus on PVR for clinical study standpoint, but again, in clinical practice, we don't check PVR that often as much as we look at in clinical trials. But again, I think it helps. Patients don't see the PVR, but it should reflect in their improvement for sure.
So it's clear that the field is kind of looking for disease-modifying therapy, and both Cereno's and Tiakis' asset has potential to emerge as one. Martin, maybe you can tell us how Treprostinil works and how it can reverse pathogenic remodeling in PAH.
Yeah, sure, gladly. And to Dr. Buddhedev's point and some of the shortcomings mentioned within his answers, with the available treatment options, we believe that our drug has some unique and advantageous capabilities as a PAH treatment candidate. And that is basically, first of all, Treprostinil is a recombinant form of an endogenous human protein I mentioned in the beginning called allophrin, which is a potent and reversible inhibitor of neutrophil elastase and other serine proteases such as proteinase 3. And based on the knowledge that inflammatory processes play a key role in progression of PAH, these unique capabilities really form the initial rationale for developing our drug in this rare disease. And in the preclinical work and within PAH-specific models, we were able to demonstrate its ability to impact two key angles of the underlying pathology, if you will. And that is, on the one hand, the inhibition of freely circulating neutrophil elastase, as well as neutrophil extracellular net formation and bioactivity. And that helped us demonstrate the ability to prevent loss and fragmentation of elastin, precapillary arterial instability and loss, as well as preventing the induction of smooth muscle cell proliferation and tissue infiltration. I mean, all these being hallmarks of the disease. And secondly, also, and this is now where we really are something of in a unique position, is that by enhancing the binding of caveolin-1 to BMPR2 signaling, so bone morphogenetic protein receptor type 2, in pulmonary artery endothelial cells, and thereby we promote downstream signaling and redistribution of BMPR2 to the cell plasma membrane, having a positive impact on BMP signaling as well, and thereby ultimately help to improve cell survival. So similar to what Sotatercept does as a ligand trap. So, and this is really a unique situation where we come at the disease from two angles. And we believe that these mechanisms collectively contribute to Treprostinil's potential to reverse the obliterative changes in pulmonary arteries and thereby ameliorate PAH.
And Rahul, what is Cereno's hypothesis around CS1's ability to reverse PAH pathology through HDAC inhibition?
Yeah, so we see ourselves as like pioneers in untapping the potential of H and using HDAC inhibition. And what is that epigenetic modulation? Actually, what it does is it's the alteration of gene expression without altering gene material. I think this is something very important to understand. And what we do is, and this has been shown through, as you were saying, HDAC inhibition, that we can reverse remodeling. We've shown that in preclinical models. There's anti-fibrotic and also anti-inflammatory effects, which in the end lead to exactly what we want to do. And that is, of course, to reduce pulmonary pressures, because it's all about reducing the hypertension. And what we see ourselves is that we are more than what Sotatercept is currently, TGF-beta modulation. We cover actually many more aspects, and we believe that with our approach, it is an oral approach once daily, and we're very well combinable with other drugs. We think we could be a very nice complementary addition to the whole armamentarium. And if one looks at the latest publication of Lancet Respiratory, where key opinion leaders, CBCT, and also the FDA had opined on what would they like to see. And as Bhuvin and Martin were elaborating on, it is really to try to tackle the root cause, and where we believe we're on a very good way.
Thank you. Martin, I know you have generated a lot of preclinical evidence on Treprostinil and how it does vascular remodeling. How does its potency compare to Sotatercept in preclinical settings?
Right, a complex question. I'll try. Maybe separate them in two parts and try to be as brief as possible. To the first part of your question, we started off focusing on transgenic mice in which we looked at the overexpression of Treprostinil, targeting the cardiovascular system to determine whether upregulation of a naturally occurring serine elastase inhibitor suppresses the hemodynamic and structural responses to chronic hypoxia. And we were actually able to demonstrate that it does in these mice. So the stress from hypoxic pulmonary hypertension is reduced in these mice. And as a next step, the viable use of Treprostinil as a PAH treatment, as well as its mechanistic role, we looked at the Sugan hypoxia rat model, still being the key model to evaluate the functionality of drugs in this disease type. And we were able to demonstrate the potential for the reversal of the vascular remodeling. And another key thing is what I find also always very important to mention in this context. I mean, translatability of animal models to the disease, especially with the Sugan hypoxia model, we had very good translatability, especially if we look at the last, 25 drugs which have been approved for markets over the last decade or so. Building on that and having demonstrated the mechanism, we looked at human tissues, and that was confirmed through use of a human pulmonary artery explant model. And so we took tissue sections containing pulmonary arteries, isolated from explanted lungs of PAH patients, and by treating them with Treprostinil, we also induced the regression of the neo-intimal changes and also the improvement of vessel lumen size. And that ultimately culminated then in our experiments looking at the insights of neutrophil extracellular traps, in short, nets, and extracellular vesicles, so in short, EVs. We looked at that from the perspective of PAH patient samples, and we were able to determine that neutrophils from patients with PAH produce and release increased neutrophil elastase and are associated with enhanced neutrophil extracellular tract formation bioactivity. And the interesting thing was then when we use and look at these IVs from PAH patients in the mouse model, and we pre-treat them with Treprostinil, we can actually prevent increases in right ventricular systolic pressure, right ventricular hypertrophy, as well as the occlusive change by the increased muscularization of the pulmonary arteries. So all in all, that gives us a very holistic preclinical picture, not just based on animal model, but also on PAH patient samples. To the second part of your question regarding the comparison with Sotatercept, yes. So what we did was compare the results we generated in the Sugan hypoxia rat model with those published for Sotatercept, and we were able to demonstrate that Treprostinil performs as good or even better than Sotatercept and that model. And that was a very exciting situation for us and really gave us the necessary confidence to engage also with the FDA, and we receive affirmative scientific advice from them for our phase 2 program. And yeah, very exciting time for us at this stage.
So you mentioned the translatability part and also phase 2 design. So how are you designing the phase 2 so that you can capture these preclinical benefits in clinical settings?
Yeah, so as I mentioned, very exciting, especially this point, and what we discussed with the FDA in particular was the ability to potentially demonstrate not just the superior clinical efficacy of Treprostinil, but also to show initial evidence for Treprostinil potentially to act as a disease-modifying therapy. And we did that, and towards the end included, so we have a generalized 24-week treatment period and included a 4-week withdrawal period at the end of treatment. So we take all patients off drug at the time point week 24. And the aim is then at the time point week 28 is to determine or to demonstrate a persisting treatment effect. And that is a key novel element of our trial design and something which was warmly received by the FDA. You mentioned endpoints. So primary endpoint is PVR, fairly typical in such phase 2 clinical trial settings. One of our major secondary endpoints is 6-minute walk distance, of course, giving us an understanding how patients feel, function, and basically how they actually perform. all in all, very similar to what has been established as a reference framework also by the Sotatercept phase 2 programs. Timelines, so we are about to have an end of phase 1 meeting at the end of this year, maybe beginning of Q1 next year, but that at the latest, and then aim for a overall program timeline of 3 years after its initiation.
And I believe Treprostinil has been previously tested in other clinical trials. What has been your experience with the drug in terms of safety profile, and are there any implications for the PAH program?
Yeah, so overall, we have used Treprostinil in 5 clinical trials so far. We have treated 109 subjects. And especially in acute indications such as coronary artery bypass graft surgery, esophageal cancer surgery, as well as COVID-19, we're able to demonstrate not just the potentially beneficial use of Treprostinil, but the overall safe use of the drug. So throughout our clinical work with the drug, we had no severe adverse events, especially in these vulnerable patient groups. And as for the implications for PAH, the safety database was really one of the foundation stones on which our communication also with the FDA was based in order to determine the transition from the acute use to the more chronic use. And based on this favorable safety profile, we did not, or basically we received this affirmative scientific advice from the FDA.
Maybe could you comment on the related positioning of Treprostinil in future PAH paradigm as you see it?
Yeah, this is really interesting. I mean, for all of us, the key question. I mean, Sotatercept will have an impact in terms of how we're going to see how it's going to shape the treatment landscape over the next few years. And of course, how do we fit into that? And I believe that based on, I mentioned the mechanistic abilities of a drug and where it goes beyond what Sotatercept can do, we have a differentiated treatment approach and mechanism of action here. So from what I see or how I perceive the drug, there's true novelty here. And in conjunction with the safety profile I mentioned before, and of course, assuming a positive further clinical development process, we see Treprostinil as a possible addition even before, not just in critically ill patients, but even in earlier stages of the disease, especially if you look at this potential for disease modification, which then could be a key game changer for PAH.
Thank you. And so, Rahul, you already ran a phase 2A trial, which was positive. Tell us more about the design and the data that you have generated so far.
Thank you very much. So we did complete a phase 2A trial. The main goal was to look at the safety and tolerability profile. Why? Because we know, and Bhuvin was elaborating to that, so far the existing drugs do have a somewhat challenging safety profile, and we wanted to make sure that we have it. We have as a basis, the first lead compound is a repurposed VPA, so there's an extensive line of experience in that, but nevertheless, we wanted to look at it, of course, with PAH patients. So we did that. And in our trial, which was for 3 months in 25 patients, we saw no drug-related serious adverse events, and it was well tolerated. So that the clinical steering committee actually advised us to stop earlier. Originally, we had intended 30 patients. We, of course, also wanted to capture efficacy signals, if there would be any. And we were very actually positively also impressed by the efficacy signals that we saw, and we also shared that in our webinars. Number one, we looked at the REVEAL risk score because we thought that is something very nice to look at, especially a small collection of patients we had. And as you know, REVEAL risk score is the most powerful risk score there is in PAH. We could see that within already the short period of 3 months of treatment, there was a large number of patients, 43%, which had a more than 1 point reduction in the REVEAL risk score. Why is that important? Because literature has shown that if within 3 months you can reduce REVEAL risk score by 1 point, that translates into a mortality reduction of 23% within 12 months. So very powerful indicator, again, a signal. We had included patients with functional class 2 and 3. And what we also saw within this time period of 3 months, that most of the patients were either stable or had an improvement in functional class. All in all, 7 patients, 5 of them from functional class 3 into 2, and 2 patients from 2 into 1. If you consider what that means, functional class 1 means that at rest, they have no complaints. Functional class 4, the worst of those, is that at bed rest, they can, even in bed, they can hardly move without complaints. So actually, a very promising signal. We had also used, by the way, in collaboration with Abbott, an inbuilt monitoring device, so-called CardioMEMS, a non-invasive monitoring device. And there we saw that approximately 2/3 of the patients showed a tendency of mPAP reduction over the 3-month period. And this is, again, something very, we find powerful. We, of course, looked also at the right ventricular function because we had done a centrally blinded echo evaluation of the patients that we had. And as you know, right ventricular function is the most important determinant of prognosis for these patients. We looked at, number 1, the so-called global longitudinal strain, which is considered to be, of course, one of the most powerful RV measurements. And what we saw was that of the patients that we had, most of them had either a stabilization or a reduction, that means improvement, I should say, of their global longitudinal strain. Not a reduction, because it is a minus value. So the more minus it goes, the better it is, to put it that way. So we saw a very nice improvement there. And this was even though the study was not statistically powered in any way, but just for the fun of it, we also looked at if these data would have been statistically significant, and they're highly statistically significant, with all the caveats, as I just mentioned. And then we also looked at tricuspid regurgitation in these patients. And here again, we saw that over 80% of the patients, either a stabilization or an improvement of tricuspid regurgitation. Again, very promising. So all of these signals, and it is very clearly signals that we are looking at and capturing, these help us also prepare for the next step, which is a phase 2b trial, which we want to embark on a global level at the beginning of next year.
So, the data's early, but looks promising, and the safety is clean so far. Are there any safety concerns with long-term HDAC inhibition?
So we are, of course, aware of VPA and the safety concerns that there are in other indications and at much higher dosages. One should mention that in our trial, we had looked at several dosages, approximately 500, a gram, and 2 grams, and we saw that the efficacy was similar. So we're most probably going to with the lower dosages. And if you take that into consideration in neurology indications, for example, one usually uses much higher dosages. So we think we're in a very good dosage arena, but we will, of course, monitor lab values, liver, thrombocytes, but also, of course, look at ECG signals, QTC prolongation, if there is any. So far, there in the phase 2a trial, we did not have any of those signs. But we will, of course, monitor, because naturally the safety of the patients, and these very morbid patients, I should say, is very important for us.
So you already touched upon the next steps for the program. Can we expect any additional analysis in the coming months on CS1?
Yes, absolutely. What we have done with this CS1 initial trial, we have also an extended access program. We should say that this will actually happen at the request of many of the investigators. So half of the patients which were in the original trial have been included in the EAP, Extended Access Program, and we are looking at those patients, following them up, and they will be investigated at baseline, 8 and 12 months, and we will, of course, share the data as they come in But here again, the primary focus, coming back to your question again, the previous one, we will be mainly looking at safety and tolerability, how these patients fare here, but we are there very optimistic. So far, no signals in that direction.
I believe you're also developing a follow-on HDAC inhibitor. I'm curious, what's the rationale behind having a similar second agent, and what makes that more suitable for IPF rather than PAH?
So what we'll do is, of course, one is a repurposed immediate release VPA that we have, but what we have also now developed is a precision deuterated valproic acid. What does that mean? That means that this has an improved profile, even though the pharmacokinetic efficacy is very similar to VPA, so we can really learn and take that experience. But what we are hoping is that the safety profile is better. Why? Because we have a reduced CYP-mediated drug metabolism. So that should improve even further the safety profile. We will, of course, look at that. And so far, the preclinical data have been very promising, and we are just about to complete the phase 1 trial that we have performed, so the SAD and MAD trial. And so far, what we hear, of course, we're awaiting the final report. It looks very good, but we will, of course, share that as the final report comes out. And that we are looking into, and we have some promising preclinical data of antifibrotic effects that we saw in the preclinical model that Martin was mentioning in the Sugen hypoxia model, but of course we are elaborating on that further.
Okay. So now that we know more about both of your assets, I would love to understand more how they're differentiated from other emerging candidates. So, for example, Insmed reported positive data from the phase 2B trial. Dr. Buddhdev, what's your high-level take on that data set, and what stood out the most to you?
Yeah, I mean, I think we kind of expected the data, right? It's treprostinil. We have known that molecule forever in various forms, oral, IV, inhaled. We know it works. In fact, I mean, it was outstanding data because it showed us the improvement in PVR and 6-minute walk distance and all of that. But I would have been surprised if it did not, because we believe in treprostinil. So, and I was surprised they actually did a randomized controlled trial. So you have patients in placebo for a known drug. Either way, I think it was great data. What differentiates them from the current Tyvaso and Liquidia's Eutrepia is that it's once a day. So I think that would be, huge for Insmed. Going from 4 times a day to once a day is probably the biggest factor. I don't think it will change the tolerability. I don't think it changes the whole concept of they're treating the disease. We are not really still curing the problem as well. So great, I think so far, but there are going to be limitations going forward, same that we are currently facing in the current treatment paradigm.
So you see it as a big improvement in terms of convenience, but maybe not so much in terms of efficacy and safety. Is that correct?
Exactly. We haven't moved the needle in that side for sure.
And do you have a preference between Eutrepia and Tyvaso?
Eutrepia just came out, so we're starting to use it. We're going to see the real world experience of it. So we have about 10 patients that we actually started upfront and seeing if they show us benefit over using Tyvaso. And if they do, obviously, we'll be leaning more towards Eutrepia. I'm excited about their print technology. I had a lot of conversations with them and how it, you can titrate the dose higher. But we want to see our own real world experience for sure on that one.
Okay. And there are some other mechanisms in development for PH as well. Gossamer has an asset, seralutinib, Pulmovant has one as well. Which candidates are you watching most closely, and what do you find exciting about these drugs?
Yeah, I would say Gossamer seralutinib probably because it's a newer mechanism of action. We would expect some degree of disease modification in that case, maybe. It's being applied for PH as well as PH-ILD, considering PH-ILD is not really looked at so much. So I'd be more excited about that. The mosliciguat, it's a similar molecule that we had it in before. Now we are using it in inhaled route. again, as we talked earlier, we're stuck in this treatment phase. We're not really changing the paradigm in terms of disease modification. So despite I'm excited about these, what I want to emphasize on, what these guys are doing, Rahul and Martin is like, I'm really excited when I'm hearing all these is they're focusing the core of the disease, right? And what we're looking forward from Sotatercept as well is that could it move up on the treatment paradigm? All the studies of Sotatercept has been done on patients who are class 4, class 3. So you look at Zenith trial, which came out. I don't know how much more information Zenith added to us on what we already knew from Stellar. But again, I think the goal was to see, could we use it up front? Could we use it as a second line, third line? And that's where we'll move the needle, where if we get these drugs out there, and we started using them up front and hit the core of the disease, it will replace the current treatment algorithm of PD5, ERAs, and all of that. And for last statement to support that theory is that when we started patients on Sotatercept, we recognized that there were some patients we were able to reduce the dose of prostacyclin. Now, was it truly the disease-modifying effect that we were seeing? But it didn't happen on everybody, and it's not all around. And it was not that we completely removed the prostacyclin, but we did reduce the dose on some of them. We'll be publishing that data in our experience soon. So that is exciting to hear that, with these molecules, we may be able to somehow at some point get rid of these first-class agents that we use right now, the vasodilators, entirety, if it's actually improving your PVR to that point.
And Rahul, how do you think your mechanism differentiates itself from all these competitors we have discussed?
I think what we have is really a unique approach in the sense, and this has been also now published extensively, not in the PH field, but otherwise, that we are really tackling epigenetic modulation venue. We do see that Sotatercept is sort of a TGF-beta trap system. We have that and much, much more. And as I was saying, what we see in the end is we see very impressive antifibrotic effects. Of course, so far all preclinical. We are going to investigate that further using some innovative technologies. We are going to, of course, look at in the phase 2b trial, what else can we do? So we think that with our mode of action, that we are adding to the armamentarium. And of course, Martin was describing another mode of action. And we are still in the early stages, but with the properties given what we know, that we are on a very good track here.
And Martin, I would love to know your take on these competitors and your differentiation as well.
Yeah, I think I can stress on really what has been said before, in the sense that if we're focusing or looking at the improvement of existing or partly symptomatic focused treatment approaches, will that translate into new treatment possibilities or even ultimately clinical outcomes? I don't know. So the key focus should really be on what we are trying to achieve in the sense of really tackling the root causes of the disease. And for us, really, it was also coming from this perspective of initially neutrophil elastase inhibition, and elafin or Buddhdev has been very well described in its role within the innate immune response system. And if we look especially at PAH patients, and was done at the PAH cohort at Stanford just recently, where we saw a significantly increase across the board. So irrespective of subtype, how long these patients had the disease, or what kind of therapy they were on, a significantly increase in neutrophil elastase and a relative deficiency of the naturally occurring protein elafin as a antagonist to the neutrophil elastase activity. So this imbalance between proteases and antiproteases is a key underlying factor of this disease. And this is something with Treprostinil, if you were to look at the clinical development arena, we're about one of a few, if not just singly, which can address these both sides. So on the one hand, the anti-inflammatory angle, but also the interactions with BMPR2 signaling. So that really sets us apart.
I would love to know what investors and clinicians should be watching for in the next 2-3 years. Rahul, you want to go first?
Sure. So I think we are hoping to naturally, as Martin also was alluding to, and Bhuvin was also saying, we have this new approach. And we think if we can tackle the root cause, we can really make a difference. We can change sort of how the right ventricle functions, not just through vasodilation, which is actually a short-term therapy. And we see that therapies have to be added on and on. And we think we could be a game changer here because we are really an anti-remodeling, anti-fibrotic drug that can make a difference and hopefully improve the lives of patients. And that's what we're aiming for and hope to show that in the next studies, phase 2b, and then, of course, the following phase 3 trial.
Thank you. Martin, is there anything else you would like to add?
Yeah, I mean, I can just second that in terms of aiming to demonstrate this potential in the phase 2 is really going to be the holy grail, showing the potential which these drugs may have and the impact on PAH. Of course, it's going to be, would be huge if that could be demonstrated. But to add to that, in short, I mean, two elements here. On the one hand, investors should or could focus also looking at novel trial designs, which provide for additional insights into the disease, be it on approach to demonstrate. We've discussed disease-modifying potential. What does that mean for a given disease? How do we actually see that from a framework perspective? Inclusion of additional live imaging technologies, MRI, CT, and also from my perspective, the development and inclusion of disease-specific development of surrogate markers and endpoints. I mean, that is the trial perspective, which could also already separate or distinguish drug candidates at that stage. And on the other hand, maybe lacking a better work, look for candidates which provide this novel ability, something which adds to the picture, not just improving on something. And as such, I believe CS1 and Treprostinil could be two good candidates to be kept on the radar.
Great. So, Dr. Buddhdev, what has been your takeaway from today's session, and what are you looking forward to in the evolving PAH landscape?
Yeah, very exciting session, and thanks for setting this up. It was incredible to hear from them directly about, their science and how excited they are, and kind of resonates with what we are hoping for changing in PAH world as well. I mean, it's so exciting that, in fact, like, I will be thrilled to be, in fact, a part of their clinical trials and sites, So I think it is very encouraging. I'm very confident that, they'll succeed. We hope for that. So great times, and we're excited for it.
Okay. A big thank you to our panelists for this insightful discussion, and also thanks to our audience for joining us. Hope you have a wonderful day. Thank you so much.
Thank you very much.
Thank you all.
Bye.